Timing of cortical genesis in the human. Since our ultimate goal is understanding the development of the neocortex in humans, the comparison of selected cellular features of the human cortex at different prenatal stages with that of the macaque monkey may help to determine the corresponding time and sequence of developmental events in these species. The determination is essential if we want to apply the findings obtained from experimental animals to the understanding of human cortical development. To this end, Polyakov's comprehensive histological studies of cortical development in human fetuses, published originally in the Russian literature, Polyakov 1959-1965, have been reviewed in more detail elsewhere, but are presented here summarized in Figure 1.6 and compared with the timing of corresponding events in the macaque monkey. Stage 1. Initial formation of the cortical plate from approximately the 6th to the 10th fetal weeks. During the 7th fetal week, postmitotic cells begin to migrate from the ventricular zone outward to form a new accumulation of cells at the junction of the intermediate and marginal zones. By the middle of this period, synapses of unknown origin are present above and below the cortical plate. This stage corresponds approximately to the level of cortical development found in the monkey fetus between E40 and E54, depending on the region. Stage 2. Primary condensation of the cortical plate through approximately the 10th and 11th fetal weeks. At this stage, the cortical plate increases in thickness, becomes more compact, and is clearly demarcated from the fiber-rich part of the intermediate zone, which seems to have fewer cells per unit volume, indicating that the first major wave of migration is almost spent. Figure 1.6. The end of this stage corresponds approximately to the E55 to E59 period in the monkey, when the majority of neurons of layers 5 and 6 are generated in most regions of the cortex. Stage 3. Bilaminate cortical plate, most pronounced during the 11th to the 13th fetal week. The uniform and compact cortical plate of the second stage becomes subdivided into an inner zone occupied mainly by cells with relatively large, somewhat widely spaced oval nuclei, and an outer zone of cells with densely packed, darker bipolar nuclei. Figure 1.6. This heterogeneity results from the more advanced maturation of the deep-lying neurons that had arrived at the cortical plate during earlier developmental stages, plus the addition of a new wave of somas of immature neurons that take up more superficial positions. This period is also characterized by the appearance of the cell-sparse, fiber-rich subplate zone situated below the cortical plate. This transient embryonic zone in the human fetus is particularly wide in the regions adjacent to the association areas. The third stage corresponds roughly to the level of development achieved in the monkey between E59 and E64. Stage 4. Secondary condensation, from the 13th to the 15th fetal week. During this period of gestation, the ventricular zone becomes progressively thinner, while the subventricular zone remains relatively wide. Figure 1.6. The cortical plate again becomes homogeneous in appearance and resembles, in a sense, a thickened version of stage 2. The reason for this change may be that in stage 4, most of the young neurons in the cortex become considerably larger as they differentiate, while relatively few new immature neurons enter the cortical plate. The result is a more uniform appearance. At the end of this stage, an accumulation of large cells appears below the cortical plate, and the subplate zone enlarges further. Depending on the cortical region, this stage appears in the monkey between E64 and E75. Stage 5. Prolonged stage of cortical maturation, from the 16th fetal week continuing well into the postnatal period. Morphological data are inadequate to determine for how long neurons continue to migrate to the human neocortex after 16 weeks, or how many do so, and hence the line at the right side of the curve is dotted in Figure 1.6b. By the 5th month, relatively few neuronal precursors seem to be proliferating in the reduced ventricular zone of the human cerebral hemispheres. However, the interneurons, which continue to be generated in the subventricular zone and the ganglionic eminence, are still being added to the cortex between the 20th and 25th weeks of gestation. A comparison of the audio-radiographic results in the monkey with comparable stages in human indicates that most neurons of the human neocortex are generated before the beginning of the third trimester of gestation. Toward term, the ventricular zone disappears, the subplate zone dissolves, and as the intermediate zone transforms into the white matter, only a vestige of the subplate cells remain as interstitial neurons. It should be emphasized that during neurogenesis and after completion of neurogenesis, glial cells are generated and migrate across the cerebral wall. This phenomenon is particularly pronounced in the human forebrain, where glial cells, including oligos, largely outnumber neurons. After all cortical neurons have been generated and attain their final position, their differentiation, including the formation of synapses, proceeds for a long time and reaches a peak only during the second postnatal year. The subject of synaptogenesis in the cerebral cortex of both macaque monkeys and humans was reviewed in the second edition of this volume, which is dated 2000. And not much new research has been done in this area since.